Welcome to the TOEFL listening section. In this part, you will hear one or more conversations in an academic or campus context, such as a discussion between a student and a professor or between two students. You may take notes while listening. Listen to part of a conversation. You will hear it only once, so listen carefully. After the conversation, you will answer questions about what you heard. Some questions will ask you about main ideas and details. Others will ask about implied meaning, speaker attitude, or how ideas relate to one another. Use both what is stated and what is implied to answer.
Good afternoon, Dr. Chen. Thanks for meeting me. I wanted to discuss my project on protein folding prediction and get your advice on methods and validation.
Afternoon, Daniel. Tell me what you have in mind. Is this for the structural bioinformatics seminar or your thesis work?
Both, actually. I plan to compare deep learning approaches, like AlphaFold style models, with more classical methods, such as Rosetta, and then apply predictions to a membrane protein case study.
That sounds promising. Start by clarifying the prediction goal. Are you aiming to predict backbone coordinates, side chain conformations, or folding pathways over time?
Mainly static tertiary structures, so backbone and side chain positions from sequence. I also want to discuss how contact maps and multiple sequence alignments contribute to accuracy.
Good. Modern models often use multiple sequence alignments to infer coevolutionary signals. Coevolution helps predict which residues contact each other in the folded state.
I read about that. AlphaFold and related networks use attention mechanisms to combine MSA data with physical constraints, but I am unsure how to evaluate model confidence.
Confidence metrics vary. AlphaFold outputs a per-residue pLDDT score that estimates local accuracy. You can also use predicted aligned error matrices to see expected coordinate errors between residue pairs.
Would RMSD against experimental structures be sufficient to claim one method is better than another?
RMSD is useful, but it has limitations. For example, a low RMSD can hide errors in side chain packing or functional sites. Consider additional metrics like TM score for fold similarity and side chain triangle deviations for active sites.
That makes sense. For my membrane protein example, experimental structures are scarce. I was thinking of using cross-link data or cryo-EM density as partial validation. Is that acceptable?
Yes, integrative validation is appropriate. For membrane proteins, you can validate transmembrane helix placement, hydrophobic surface exposure, and compatibility with known cryo-EM density. You can also test whether predicted structures respect known disulfide bonds or post-translational modifications.
One concern I have is how training data bias affects predictions. If the training set contains many soluble proteins, can models generalize to membrane proteins?
Training bias is a real issue. Models trained primarily on the protein databank are influenced by the composition of that database. Membrane proteins are underrepresented in PDB, so specialized fine-tuning or including homologous sequences from metagenomic data can help.
So adding diverse MSAs and perhaps transfer learning could improve membrane protein predictions. That seems doable. What about folding dynamics versus static predictions? I know molecular dynamics simulates motion, but is expensive.
Exactly. Neural networks give static models quickly, while molecular dynamics can explore folding pathways and stability, but at high computational cost. A hybrid approach can work, using predicted structures as starting points for shorter MD runs to assess stability and refine side chains.
I like that. In terms of architecture, should I implement an end-to-end transformer-style model or focus on a contact prediction network followed by energy minimization?
It depends on your resources. Transformer-based models can capture long-range interactions directly, but need substantial compute and training data. A two-stage pipeline with a coevolutionary contact predictor and subsequent physics-based refinement is more accessible and still competitive for a study.
For evaluation, I will include CASP-style benchmarks. Are there specific CASP outcomes I should highlight?
You should definitely discuss CASP 14, where AlphaFold demonstrated near experimental accuracy on many targets. Mention how improvements in residue pair representation and end-to-end differentiable structure modules contributed to that performance.
When you say near experimental accuracy, do you mean the models matched crystal structures exactly?
Not exactly. Near experimental accuracy means that predicted coordinates often fall within low RMSD ranges compared to solved structures for many targets, and that sidechain packing and active site geometry were correct enough for many downstream applications.
That clarification helps. I plan to include a case study where a predicted structure guided mutagenesis experiments. Are there examples I can cite?
Yes, there are studies where AlphaFold models were used to design stabilizing mutations and to propose ligand binding modes when no experimental structure was available. Cite those as examples of practical applications in drug discovery and enzyme engineering.
Perfect. Finally, any suggestions on reproducibility and reporting so reviewers can follow my workflow?
Document your training and test splits. Specify MSA sources, list software versions, and deposit predicted models with metadata. Include confidence maps and validation metrics like per residue, pLDDT, TM score, and RMSD where applicable. That will make your work transparent and reproducible.
Thank you. This gives me a solid plan. I will draft a methodology section and bring it to our next meeting, along with preliminary results.
I look forward to it. Remember to be explicit about limitations, especially regarding training data bias and cases where predictions may be unreliable. Good luck, and send me your draft when ready.
Listen again to part of the conversation. Then answer the question.
Near experimental accuracy means that predicted coordinates often fall within low RMSD ranges compared to solved structures for many targets, and that side chain packing and active site geometry were correct enough for many downstream applications.
This concludes the conversation portion. For more TOEFL practice tests and study materials, visit Spinta.org.